seeing is only a small part of perception. Deciphering, decoding the image of a well-camouflaged moth, for instance, requires pathways beyond the striate, or primary visual cortex. So too these random spots, bars, edges, and visual frequencies give us a sense of size and shape, but they tell us little about Dalmatians. Higher visual areas in your brain are now at work, a region of near unexplored brain territory to which Dr. Mortimer Mishkin has devoted much of his research.
The striate cortex is simply the first station in the cerebral hemispheres to receive information from the retina. And it looks as though the striate cortex in the back part of the brain sends information forward along two different pathways. One headed to the top part of the brain, to a region that we call the parietal lobe, and another headed toward the bottom part of the brain, toward a region that we call the ventral part of the temporal lobe.
That higher pathway, plunging in and out of the exterior cortex, makes a series of very specific visual computations. Its role is to tell us where things are in the visual field, to specify that the dog is in front of the mother and child, to establish that this couple faces the faraway lake. Clearly, this pathway helps us orient one object to another, a visual back and forth that continues as long as our eyes remain open. The lower pathway's function is possibly the most surprising of all. In a similar series of stages, it helps us explain what objects are. It tells us that this is a dog, this is a child, this is a man. As neuroscientists investigate our visual system, they find that our brain cells here are very, very specific. In the 1950s, some neuroscientists called them grandmother cells. The theory, which enjoyed a certain popularity, was that we might actually possess a single cell that lit up when our grandmothers came into view, and another cell for each and every person we ever saw. These cells were pictured as being almost miniatures of that person. However startling the theory, it now seems that these cells at the end of the lower pathway actually might be similar to grandmother cells.
Some of my colleagues working in Princeton and at Oxford University have recently shown that they can record from single neurons in part of the inferior temporal lobe, and these neurons will be activated by faces better than by any other kind of stimulus that they can present. Not only will they be activated by faces, but if the faces are distorted in certain ways, the neurons will not respond.
As we watch, we can hear single cells in the inferior temporal lobe of monkeys reacting to a single monkey face. The cell only fires if the face is at the right angle and is not distorted. What distinguishes these very specific cells from the so-called grandmother cells of the 1950s is that they rely on many other cells in the visual pathway to feed them detailed information. They're called cumulative cells. And so, grandmother is not merely embodied in a single cell. She's represented in our brains by a multitude of cells, some so simple that they merely analyze the bars and edges of her face, the visual frequencies of her complexion, and some so complex that in the lower pathway of vision, a single cell might actually light up when we see our own grandmothers, but only with help from the hundreds of thousands of cells that precede it. Utilizing modern brain research techniques, Dr. Leslie Ungerleider, a colleague of Dr. Mishkin at the National Institute of Mental Health, has been studying the process of human facial recognition.
Until recently, our understanding of how the brain codes visual information has come mainly from studying the activity of single neurons in the monkey brain, and also by looking at impairments in patients following brain damage. But now, with advances in the new technology of functional magnetic resonance imaging, we can look directly at activity in normal, healthy individuals' brains, and we can see how that activity changes as these human subjects are involved in various kinds of tasks. We're especially interested in what happens in the brain when people perceive, recognize, and recollect faces. Just as we had found in our monkey work, our imaging studies in humans indicate that there are specialized regions in the temporal lobe of the human brain that appear to be specialized for the processing of faces. When we see faces, these areas of the cortex become active. Imaging studies have also shown that beyond the visual cortex, there are areas in the frontal lobe that are involved in the processing of faces. In particular, these areas become active when we see a face and then must hold that face in mind when it's no longer present in order to recognize it a short time later. Studies such as this tell us that areas of the frontal lobe are involved in maintaining these short-term memories for faces. We also have evidence that the very same areas of cortex that are involved in the processing of information about faces also participate in the long-term storage of memories for faces for subsequent recollection. In fact, when we imagine a face in our mind's eye, these very same areas of cortex also become active.
Higher vision is not limited to faces. It may help pick out the one lame animal in a herd of 25,000. For the lion, such visual computations are everyday tasks. Could he meticulously examine all 100,000 legs for wounds? Or does he look for visual exceptions, for an irregularity of movement that stands out more clearly than a bloodied leg? How do we spot an old friend walking away from us in a crowd? By the color of his socks or by the way he moves? Either way, while we call this process seeing, it's a very high order of seeing. It's perception. And how long does it all take to happen? Well, the process of seeing and perceiving something as elementary as a flash of light takes about 3/10 of a second. There, that's vision. Blink your eyes and you might have missed it. Slowed down, the route of vision is clearer. First, we can see the back of the brain light up, here in the area studied by Hubel, Wiesel, and de Valois. Now, with images being computed along those pathways of Mortimer Mishkin, the collective information reaches the front of the brain, the thinking part of the brain. And it's now when we realize what we've just seen.